Hello students, welcome to CBC classes. In previous session we have learned about innate immunity, and in this session we are going to discuss primarily about acquired immunity. Acquired immunity is specific immunity, which means the immunity that is against specific pathogens. That is the reason acquired immunity is called as specific, whereas innate immunity was non-specific. And we have different types of examples we have discussed in innate immunity, like physical barrier, which consists of skin, mucous membrane, right? And we have learned about various other examples of innate immunity, whereas that is chemical cytotoxic barriers, or we have physiological barriers which secretes, the membrane secretes various secretions like mucus or saliva. The saliva consists of lysosomes which kill the bacteria. Okay, mucus which prevents the movement or mobility of the various foreign substances. And we have learned about the cells, WBC cells, that is neutrophils, mast cells. We have learned about macrophages, and we have learned about interferons. Interferons are cytological barriers. They are categorized under cytological barriers. The viral infected cells were producing interferons, and these interferons were attached to the normal cells and preventing the viral infection to grow to or to spread to the other cells. That was the role of interferons that we have discussed in our previous session. And we have learned about macrophages, or we have learned about neutrophils. What they do, they generally phagocytize. And we have also discussed about natural killer cells in innate immunity, which used to secrete certain substances that used to kill the bacteria or viral or any other foreign substances, right? That is what we have learned about innate immunity. In today's session, we are going to learn about acquired immunity. In this, we have two types, that is active and passive. Active and passive acquired immunity that we are going to discuss. Active further classified as natural and artificial, and passive is also categorized as natural and artificial. Okay, we will learn the difference between active immunity and passive immunity. Okay, before that we will also discuss about inflammation. Inflammation is the body response or body defense response for injury or for any spread of the bacteria. It stops, it eliminates the bacteria, or it stops the growth. Okay, so this is inflammation. You must have heard about inflammation in our layman language also: swelling, pain, tenderness. Okay, we have a swelling over here. Mom, I fell down, then I had a swelling in the knee. Okay, I have a pain in the knee, and this is the knee injury turned into reddish in color. So these are all signs of inflammation. Inflammation is also non-specific, so it comes under innate immunity. So in innate immunity, we have learned about physical barrier, we have learned about physiological barrier, we have learned about cytological and cytokinin kind of chemical barriers and cytological barrier, right? Now we are going to learn about inflammation. After discussion about inflammation, we are going to learn about acquired immunity, which has two types, active and passive. So in this session we are going to learn about acquired immunity, and we are going to learn about the difference between active and passive immunity. This is very important topic, important in the point of NEET questions. So I am going to highlight those areas where the questions can come. So let us learn about inflammation. Inflammation is, see, whenever you have an injury or growth of bacteria or any pathogen. For suppose, let me take as a pathogen so that it can categorize every organism: viral, fungal, protozoal. Okay, what happens? Our body shows certain signs. Body shows certain signs. These signs are basically swelling, edema. We also call it swelling as edema. You can call it as swelling, edema. Okay, and we have other signs like pain. Okay, redness. Third, redness. So pain, redness and swelling. What else? What else you can feel when somebody is injured or some swelling can be seen? Okay, pain, redness, swelling, and any other symptoms that you can show? You can tell or you can discuss. Any guesses for inflammation signs? Yes, edema. What is edema? When there is an injury or where there is a external force that is applied. For suppose, let us take an example. An external force, somebody punched on your hand, okay, with a large force, not with the small force, large force. Okay, what happens? The tissue over here, okay, undergoes swelling. Okay, there is a swelling of the tissue. Vasodilation happens as a result. What do you understand of vasodilation? The blood vessels that are present near the injury, they swell. They cause pumping of more amount of blood to those areas where the structure is inflamed or injured. Okay, this is the swelling. Then you can feel pain, also called as dolor. Okay, and this redness, which is rubor, edema, because of injury impact and because of vasodilation, more amount of blood is going to that particular area. What happens? That particular area appears red in color. So redness, rubor or edema. These are basically general signs that you notice during inflammation. Okay, and the fourth important symptoms that everybody face is fever, raise in the temperature. So wherever there is a injury or any pathogen that is entering the body, the first reaction of the body is fever. Now how fever is controlling the growth of the bacteria? Because it is increasing the body temperature, the suitable temperature that is required for the pathogen to multiply is no more. So the bacteria multiplication or pathogen multiplication will stop. This is how the fever is helping controlling the pathogen growth. And when there is an increase in fever, the pathogen requires certain elements or certain minerals to replicate, like iron. For example, let us take iron is required. And when the fever is there in the body, what happens? The iron availability to these bacteria decreases. Okay, because increase in temperature, the availability of iron decreases. As a result, the bacteria is no more able to replicate. So here you have to remember the four signs of the inflammation, that is swelling or edema, pain, that is also called as dolor, redness, which means rubor, and we have another symptom, that is fever. Okay, so fever is the one which controls the growth of the pathogen. Okay, so this is also a sign of innate immunity. Inflammation is a sign of innate immunity. So in inflammation you can see these four signs. So it is a natural mechanism or natural mechanism of defense system. Here, what are the signs of inflammation? You have to remember these signs every time. Okay, the very first sign is fever, raise in temperature. Fever, which means raise in body temperature. You can measure the body temperature through thermometers that you must have learnt in your smaller classes, right? When you are in 7th or 8th, you must have learned that fever can be measured by thermometer, right? Now after fever, swelling, okay, is the second sign. This causes vasodilation. More amount of blood reaches to the site. When more amount of blood reaches, blood consists of various cells like neutrophils, okay, WBCs, and WBCs or platelets or immunoglobulins reach the swelled site of the inflammation and they fight against the growth of the pathogen. Now coming to fever, swelling, redness, again this is also because of vasodilation. So because of vasodilation, more amount of blood is reaching to that particular area. That is the reason that area appears to be red in color. Then the fourth sign is pain. Because of the swelling, we have, or because of the damage to the tissue we have pain, because the tissue is having nerve fibers, and these nerve fibers caused by the swelling puts pressure on the adjacent tissue. As a result, the pain can be felt, and this pain can be felt because of the nerve fibers. So these are the signs of inflammation. Now let us learn about acquired immunity. What is acquired immunity? Innate immunity was present by birth, but this immunity is not present by birth. It is formed against specific pathogen, specific bacterial or viral particles. That is, then it is called as acquired immunity. So here this is specific for pathogen. Okay. The first response of the body, which means, let us understand the first response of the body. Okay, this is response of the body I am talking about, and second response of the body. Okay, the first response of the body over here. When a bacteria, first suppose I am talking about a bacterial infection. Now let us see, a bacteria is entering a body. Then our body consists of different types of cells. Innate immunity is there, right? It will prevent the bacteria. Then if suppose it has crossed the innate immunity and it has entered some particular body tissue, for suppose muscular tissue of hand, okay, and an injury over here, and the bacteria has entered, somehow entered to the this muscular tissue. Now what is the response of the body? The lymphocytes are produced. Which type of lymphocytes? B lymphocytes and T lymphocytes. There are two types of lymphocytes, B lymphocytes and T lymphocytes. That about B lymphocytes and T lymphocytes we are going to learn in our coming classes. Okay, for now just understand, B lymphocytes are the one which form the antibodies, and T lymphocytes have helper T cells, killer T cells and memory T cells, which keeps the memory of the bacteria that is entering the body. Okay, so basically T lymphocytes have memory cells, memory T cells, which remember the structure of the bacteria that is entering the body. We have different types of bacteria, we have different types of viral infections. Name them, name some bacterial infections. Okay, what are the bacterial infections? Tuberculosis is a bacterial infection, right? Any other bacterial infection that you want to give? Diphtheria, okay, tuberculosis, leprosy, all these are different types of bacterial infections, right? They cause certain immune reaction, because when they are entering into the body, the immunity inside the body will respond. When it enters for very first time, for suppose tuberculosis is entering, Mycobacterium tuberculosis is entering the body, or Vibrio cholera is entering the body. Okay, the first response, what do you understand by the cholera is entering the body for the very first time, right? Okay, then our body will react. Okay, that is what response is. The body will react to bacterium that is entering, right? The body immunity will react to the bacterium that is entering in the body. How? By forming T lymphocytes and B lymphocytes. By forming T and B lymphocytes. T lymphocytes have memory and B lymphocytes produce antibodies. The structure of antibodies, the role of antibodies, that we are going to learn in tomorrow's session. Just remember, T lymphocytes and B lymphocytes are formed. But because this bacteria is entering in the body for the very first time, the T lymphocytes and B lymphocytes that are formed are slow process, are formed in a slow process. Because the structure of the bacteria has to be recognized. According to the structure, antibodies have to be produced, T memory cells have to be produced, or B lymphocytes have to be produced, right? It takes time. Okay, so this response, first response of the body towards this bacteria will be slow. We can take example of viral also. Many viral infections, like common cold is a viral infection, right? Or COVID is a viral infection, SARS is a viral infection. So viral infection, if viral pathogen is entering the body takes certain time to respond towards the viral growth or bacterial growth. Okay, so in this time T lymphocytes are formed, B lymphocytes are formed. Okay, so this is a slow process. This is called as first response of the body. Now coming to the second response, same bacteria, for suppose in first response I have taken example of cholera. Okay, second time the cholera bacteria has entered after few years, after 1 year, after 2 years, the bacteria has re-entered the cholera bacteria has re-entered the body system through contaminated water or food. Okay, now what happens? This time our defense system is active. It has a memory. T lymphocytes has a memory about this bacteria. It will form antibodies very fast, very quick, and it will eliminate the bacteria. Okay, so second time the cholera bacteria is entering the body, what happens? We have already T memory cells. Okay, memory of this particular bacterial structure. What happens? The T memory cells recognizes the cholera bacteria and immediately will inform to the B lymphocytes and killer T cells. Now this killer T cells and B lymphocytes will produce antibodies. B lymphocytes will produce antibodies and killer T cells come to the cholera bacteria. Killer T cells and antibodies formed from B lymphocytes naturally kill the cholera bacteria very fast. So second response is very fast and specific. They easily recognize the bacteria or the pathogen that is entering the body and they kill those organisms. So this is the first response of the body and this is the second response of the body. Okay, so this is acquired immunity. This is immunity for specific pathogen. The antibodies that are produced for cholera cannot kill the pathogen of other bacteria. For suppose if cholera bacteria have certain set of antibodies, these antibodies will kill only cholera bacteria. It will not kill typhoid bacteria or it will not kill poliovirus. So every species has specificity. Okay, so these are the characteristic features that we are going to discuss now. Now you have understood the first response of the body and second response of the body, which is faster. Any guesses? You don't have to guess, we just understood the scenario over here. The first response is always slow, second response is always fast. The immunity has a memory. The memory is because of T cells. T cells are further divided into killer T cells, memory T cells. Okay. Now B lymphocytes form the antibodies to kill those infectious bacteria or infectious pathogen. Now let us see the characteristic features of the acquired immunity. Just now we have discussed what is acquired immunity. Now we will see the characteristic features. Specificity, species, specificity. These form antibodies, T lymphocytes and B lymphocytes, or you can say immunity. In short term you can use immunity is specific to pathogens, which means The T lymphocytes and B lymphocytes, if they are formed against some virus, polio virus, okay, then it will be for polio virus only. It will not act on the smallpox or chicken pox. Okay, so they are specific. Then comes diversity. Now what do you understand by diversity? Just now we have understood a diversity. That means there are different types of microorganisms, then different types of T and B lymphocytes will be produced. Different types of immune response will be there for each microorganism. So this is called as diversity. So diverse microorganism, which means there are different types of bacteria, virus, fungi, protozoa, right? Diverse microorganism, which means diverse immune response. Right? The third point over here, that our immunity can identify the self and non-self. Now what do you understand by self and non-self? So these tissue, these cells, lymphocytes, B lymphocytes or T lymphocytes can identify our own tissue, and non-self, which means bacterial infections or viral infections, they can identify which is self tissue. Otherwise, if it is not able to identify, it can kill our own cells, our own tissues. Okay, so these B cells and T cells, T lymphocyte cells, can only kill pathogens. They don't harm host tissue. They don't harm host tissue, host body. So self and non-self, they can differentiate self and non-self, and B T lymphocytes only kill the pathogens. So these are the important points that you have to remember about immunity. And the immunity response is specific, I agree, but it is first response will be slow, and if the response, if the bacteria is again infecting the body, for the second time the response will be faster. This technique is used in immunization or vaccination. We use vaccination term very widely, right? So what is vaccination, that we are going to learn now. Okay, so acquired immunity is basically, in the first response it will be very slow because the antibodies have to form, B lymphocytes have to form, then only antibodies will be forming, and then T lymphocytes have to form. These T lymphocytes have M-T cells, which means memory T cells, which will keep the memory, which will keep the structure of the antigen, that is antigen of the pathogen, right? So this is the response. First will be slow and second will be the fast, because this technique can be used in the immunization. Now how vaccination work? Let us see the vaccination process. Vaccination, the pathogen. Initially what happens, the dead pathogen, which means the pathogen is killed. For suppose let us take a bacteria, a virus, polio virus, okay. In polio virus, what it causes? Polio virus causes paralyzation of the lower limbs, okay, or you can say undergrowth of the lower limbs. The lower limbs don't grow with the age, okay. As a result, the person limps, right? So this polio virus was very deadly. Now how it was stopped or it was eliminated from the India? Through vaccination. What they did is they took small sample of polio virus live and they killed the polio virus through heat. Okay. Now whatever the virus is present, it is a dead virus. So if the dead virus is entering the body, it will not cause infection, right? If this dead virus is entering the body, this was a polio virus. Let us take in a structure, some typical structure of the virus over here. Okay, this is heat killed, which means it will not cause any disease. The pathogenicity of the virus has been removed. Heat kill virus enters the human body. Okay, and human body have defense mechanism. What are these defense mechanisms? Primarily in the acquired immunity I am talking about T lymphocytes and B lymphocytes. Okay, now slowly these T lymphocytes and B lymphocytes will come to the virus. Okay, they will scan the virus. The virus is dead, it is not causing any infection. They will scan, they will see the antigens that are present, okay, the protein structures that are present outside the virus. The virus is having a protein code, right? This protein code is called as antigen. Okay, now these T lymphocytes and B lymphocytes will scan this viral particle and will slowly multiply. Okay, they form killer T cells, memory T cells, which means these memory T cells will keep the memory of this virus. Okay, killer T cells are formed, and what else? B lymphocytes will produce antibodies, or you can call it as immunoglobulins, antibodies, or Ig, which means immunoglobulins. We are going to learn about immunoglobulins in our next chapter, so I'm going to explain what is Ig or antibodies. Now these lymphocytes and these antibodies and killer T cells will kill the virus. Already it is not there, but it will remove entirely. Okay, and memory T cells are present. This is our first response. Now in future, if the person is infected with the real polio virus, live polio virus, what happens? What is the second response? Immediately the memory T cells will come in contact with the polio virus and it will recognize the structure. Oh, this is the danger. And we have already formed immunoglobulins, we have in the body, and already formed B lymphocytes in the body, right? Already formed the killer T cells in the blood or in the body tissue, right? What they will do? They multiply rapidly and they will reach to the polio virus and they will completely kill the viral virus before its replication, before it can show or before it can affect any other body structure. So this is the second response. So this is how the acquired immunity is used for vaccination. So in the very beginning of the classification of immunity, we have seen acquired has been classified into active and passive. We are discussing about acquired active immunity, right? This active immunity has artificial and natural, right? So artificial form of acquired immunity is immunization. This is immunization. If you give, this is the example of immunization or vaccination. Okay, vaccination. This principle has been used for vaccination. So what they do throughout the vaccination, whenever you are getting TBT vaccination, tetanus vaccination, okay, any kind of vaccinations, polio vaccination you must have heard about. These type of vaccination, what are these? They basically create an artificial defense system in the body. The first response is how these liquids or some samples of vaccine consist of dead particles of the virus or dead bacteria. They insert the immunity, the memory T cells form a memory of this dead virus or dead bacteria, and after, if the person really gets this infection from another person, what happens? The immunity is triggered and it will kill the pathogen very quickly. So this is used in the vaccination. This is how polio is completely eradicated from India. Smallpox has been eradicated from the India. Now no more smallpox patients can be seen, or no more smallpox or polio virus patients are seen. Okay, so this is natural. We have learned about natural system. So whenever the person is infected from one kind of infection, he cannot be infected by another kind of infection or same kind of infection twice, because our body has immunity. Then ma'am, why we are getting cold or COVID multiple times? Cold and COVID, these viruses, they have genetic changes very fast. They mutate very fast. The protein coat of the common cold virus or virus of COVID, certain viruses or certain bacteria change very fast and they multiply very fast. As a result, the immunity that is present to the earlier variant will be not useful for the new variant. As a result, we are getting common cold very frequently. We get certain infections very frequently. Why? Because these viruses or these bacteria change very frequently. Now coming to the, now we have done with the vaccination. Let us understand between active immunity and passive immunity. Okay, we have learned about acquired immunity. Now we have active immunity, which means, and passive immunity. Active immunity, our body has responded, the first response and the second response that we have discussed is called as active immunity. That means the immunity that is developed by our own body, our own defense systems, or by T lymphocytes and B lymphocytes, then it is called as active immunity. Okay, what do you understand by active immunity? That immunity response developed by our own body. Then this is called as active immunity. Now coming to passive immunity. What do you understand with passive immunity? So whatever we have learned, the first response and the second response of the immunity, that is generally active type of immunity in acquired immunity. Now coming to passive immunity, this is a ready-made immunity that we are getting to the body. Like our body system is not working to form those antibodies. Here in active immunity, B lymphocytes were forming the antibodies, but this in passive immunity, the antibodies we are getting from outside, and these outside this outside antibodies are killing the pathogen. For example, colostrum. Now what do you understand by colostrum? When mother gives birth to child, okay, the milk, that is the first milk that comes from the breast of the mother, is yellowish in color. This yellowish color of the milk, the first milk, is called as colostrum, which consists of IgA antibody, So when the infant sucks the milk, he gets the colostrum, right? This colostrum consists of antibody A. This infant is protected from various kinds of infections because of antibody A or immunoglobulin A. Ig, which means immunoglobulin A. This immunoglobulin A protects the infant from various kinds of infection. So this we are getting, the infant is getting from mother's defense system, right? So this is called as passive immunity. This immunity is not developed by our body, we are getting from the outside, then it is called as passive immunity, like colostrum. Okay, because infant is premature in defense system, it doesn't have active defense system or active immunity. This infant cannot produce antibodies, right? So it is getting immunity from its mother through mother's milk. That is the reason mother's milk is very important to the infant in initial days, because in initial days the milk is called as colostrum, which is yellowish milk, because the yellowish milk is because of IgA antibody. So this you must remember. This is a very important NEET question actually. Colostrum consists of which type of antibody? IgA antibody. This kills the virus or your bacteria, fungal infections that are developed in babies. Okay, so this is passive immunity. Another example of passive immunity, placenta. Now, what do you understand placenta? This is a placenta connection is present between a mother and unborn child. Okay, in the uterus. So it has a connection that is called as placenta. This placenta provides nourishment to the developing embryo, right? Developing fetus, it requires energy, it requires oxygen, it requires glucose, it requires amino acids so that it can grow, right? And the excrete substance has to be removed. This is done by placenta. Now placenta also protects the fetus, developing fetus, from various kinds of infection. How? Because this placenta is also sending some antibodies to the fetus. The antibodies that can cross the placenta are IgG. Okay, again this is very important. IgG can cross. So placenta is just like a barrier that is present between mother and fetus, can cross placenta and reach to the fetus, developing fetus. So what happens? The fetus is also protected from various kinds of infection. So when the baby is born, he naturally had IgG antibody. There are 5 types of antibodies: IgA, IgG, IgM, IgE and IgD. These are the 5 types of antibodies that we are going to learn. Okay, IgA is present in colostrum. IgG is the one which crosses the placenta. These are the characteristic features that you have to remember. Again, this is a neat important question. IgM is the first antibody that is formed against the bacterial or any kind of infection. IgE is formed against allergic reactions, okay, and which is present on the cell membrane. About immunoglobulins or antibodies or types or structure, we are going to learn in tomorrow's session. In this session, we have learned about acquired immunity. Acquired immunity, that is specific to the pathogen. The immunity that is developed in the body, it is specific to the pathogen. Innate immunity, it was not specific to the pathogen. Right? Neutrophils can kill any type of bacteria. It is not specific type of neutrophil is present for specific pathogen. Okay? So here if you see the difference between acquired and innate, innate is present by birth, but acquired is acquired throughout the life. That is the reason it is called as acquired. Okay? Now acquired is further classified as active and passive. Active, which is developed by the body. Passive, we are getting from external source like mother's milk, from placenta. Okay, in passive also there is a natural. This is the placenta and colostrum is an example of passive immunity. Passive, natural immunity. Whereas there is another type, artificial, which means if the patient is suffering from any kind of infection, he has to recover, acute kind of infection, he has to recover. What we give some antibiotics, we give anti-serum, right? So this serum consists of these active immunoglobulins which will specifically kill those bacteria. Okay, these are not specific, whereas active one are specific. Okay, they are fast, passive one are very slow in action. So these are the common differences that are given in your exercise in your book sheets. So please read those differences between active and passive. The main difference is active is developed by our own body, and passive that we are getting from the external source. Like mother, natural ways, the colostrum and the placenta are the examples, and in artificial, antibodies or anti-serum is given to the acute suffering patients. Okay. Now coming to active immunity, this is developed by our own body systems. And this concept is used for immunization or vaccination, which is a artificial system of active immunity. So we have discussed about active and passive immunity, and we have discussed about acquired immunity. In tomorrow's session, we are going to discuss about antibody. Antibody is made up of polypeptide chain. 4 types of polypeptide chains are present, 2 heavy chains and 2 light chains. These heavy chains and light chains are basically peptide chains. Okay, they have amino acids. Right now, H2L2 is the formula for the antibody, and these antibodies are linked, these light chains and heavy chains are linked to each other by disulfide bonds. Okay, they have antigen binding sites from which these antibodies are binding with the antigen and they are destroying the antigen actually. So this is a antibodies function, right? We are going to learn about the structure and we are going to learn about types of antibodies, and we are going to briefly discuss about their roles. Then we are going to discuss worksheet. So today we are done with active and passive immunity. Let us discuss worksheet 7 questions over here. So here we are discussing active immunity and passive immunity questions, and we are discussing about immunity questions. So let us discuss first question. The physical barrier preventing pathogen entry is. So this is innate immunity, and innate immunity it consists of physical barrier, physiological barrier and cytological barrier. Okay, so what is the physical barrier? It is just basically a layer, okay, which is preventing the cross of the pathogen into the body. Okay, so this is a epithelium tissue is a, yes, physical barrier. Okay, mucous membrane is a physical barrier. So skin is an example of physical barrier because it is not allowing any pathogen or pathogen that is present in the environment not allowed inside the tissue, because this epithelial tissue is tightly bound to each other. It doesn't have any intercellular spaces and it is a dead tissue. The outermost layer is dead tissue. So bacteria cannot enter or fungus cannot, or any protozoal infection or any kind of pathogens cannot enter the body. Okay, so here the skin is the correct answer. The physical barrier that is present, okay, is skin. Neutrophils, plasma cells and RBCs. So plasma cell, neutrophils, if you see, they are coming under cellular barriers. They come under cellular barriers, and what they do, they phagocytize. Okay, and what about RBCs? They have different role. They carry oxygen. They don't provide any kind of immunity, not innate or acquired. Same with the plasma cells. Now let us go to the next question. Question number 2. Which type of cell helps fight infection by swallowing foreign particles? Let me erase the board. Okay, here type of cells fight infection by swallowing foreign particles, which means phagocytosis is happening. This is seen in WBCs. WBCs is a word that is used for large different types of cells. Okay, white blood cells. Now white blood cells are classified as granulocytes and agranulocytes. Granulocytes consist of neutrophils, eosinophils and basophils, whereas agranulocytes consist of macrophages, okay, mast cells. So phagocytosis is done by certain types of WBCs like neutrophils. They kill the bacteria, they engulf the bacteria, they digest the bacteria and they excrete the remnants. So this process is done by neutrophils, macrophages, okay, mast cells. So the answer over here is going to be the white blood cells. Now coming to the red blood cells, they carry the oxygen and carbon dioxide for exchange of gases. Platelets, what the platelets are doing? They clot, they are helpful in clot formation in injury. Neurons, they carry the signals. So all these three answers are the not correct option. Here the foreign particles are engulfed or swallowed by white blood cells. Okay, now moving to the next question, question number 3. Which of the following cells are phagocytic in nature? Just now we have discussed phagocytosis, which means the cell, if this is a cell and this is a viral particle that is present, okay, this cell just basically engulfs the viral particle by forming pseudopodia, and now slowly it is engulfed. Okay, now what happens? This viral particle is digested by lysosome, okay, hydrolytic enzymes, and it is destroyed, it is killed. So this is killed intracellularly. This type of process is called as phagocytosis. This is done by neutrophils. Okay, so here the answer is going to be neutrophils which perform phagocytosis. Whereas coming to the T cells, these are T lymphocytes. They have killer cells, memory cells. They induce B lymphocytes to form more antibodies. Okay, coming to platelets, we have discussed again, they are for clotting of the blood. Coming to erythrocytes, they are RBCs, red blood cells, which are helpful in carrying of oxygen and carbon dioxide and exchange of gases. So here the answer is going to be the neutrophils. Now going to the next question, question number 4. What is the name of the protective response that causes the body temperature to rise? The protective response that causes the body temperature rise is inflammation, fever, allergy or immunity. The protective response, rise in temperature, which means the normal temperature is near about 98.7 degrees Celsius. If the 98.7 degree, 97.8 degree Fahrenheit, not Celsius, okay, Fahrenheit. Now this is the normal temperature. When the normal temperature elevates, which means it becomes 99 or 100, then there is a rise in temperature which is called as inflammation, which is called as fever, which is a sign of inflammation. So here the answer is going to be the fever. Now coming to the next question. Question number 5. Which of the following is example of chemical barriers? Chemical innate immunity. So we have discussed about innate immunity in our previous class, right? So here chemical barrier they are asking. They are not asking about physical barrier. Skin is a physical barrier. Mucous membrane cilia is a physical barrier. Whereas lysosome is a chemical barrier. Why? Because these cells secrete enzymes, lysozymes. Okay, they have lysozymes, hydrolytic enzymes which kill the pathogen. So this is a chemical barrier. Now coming to the next question, inflammation is caused by release of. The inflammation is caused by release of cytokines. Yes. Now what do you understand by cytokines? Cytokines are released by cells, okay, intracellular, extracellular they are released so that other cells get, like B lymphocytes and T lymphocytes get activated and they secrete antibodies. That is the role of cytokines. So whenever cells are infected, they release certain substances so that they can activate other cells to form the, so they are intercommunication, they are used for intercommunication to activate other cells regarding the bacterial infection. So the answer over here is cytokines. Now moving to the next one. Question number 7. Antimicrobial peptides are primarily produced by skin cells, B cells, T cells or neurons. Antimicrobial peptides are primarily produced by skin cells, okay. So they kill the bacteria. So these peptides are present in saliva or any other secretions, sweat secretions, okay, sebaceous secretions or mucus secretions. So here the answer is going to be skin cells. Now coming to the next question, question number 8. Which process involves engulfing and digesting of pathogens? Engulfing and killing of pathogens, we have discussed the diagram earlier in previous questions. That is, they are forming pseudopoietic extension, then they form the food vacuole. In this food vacuole, the pathogen is present. They are digested by the lysosomes. Why? Because the lysosome consists of hydrolytic enzymes which break down the pathogen, trapped pathogen, okay. So this process is called as phagocytosis. Now phagocytosis, you understand the meaning of phagocytosis, yeah, then you can obviously mark the correct answer. Now what about endocytosis? That is removal of substances, and cytokinesis, which is a chemical exchange between cells regarding or relating inflammation. Now coming to the next question, question number 9. Interferons primarily defend against. So interferons are produced from viral infected cells, normal cells. Now these normal cells are infected by virus, right? Now these cells, will secrete certain chemicals, okay, and these chemicals are called as interferons, which bind with the normal adjacent normal cells, and they change the structures. They bring a cascade of protein changes in the membrane or within the cellular structures of the other normal cells. As a result, the viruses cannot replicate or cannot infect other healthy bacteria or other healthy cells. Okay, so interferons are primarily defense against viral infections. So these are commonly seen in only viral infections. They are not seen in fungal, or they are not seen in protozoal or bacterial infections. Okay, so interferons are different types of interferons are present for different types of viral infections. So they are again specific to the species. Now coming to the 10th question, assertion and reason. Fever helps inhibit pathogen growth. As I've told you, increase in temperature, what does it cause? It will prevent the pathogen to multiply, because the bacteria or virus needs appropriate temperature to multiply or to grow, right? If the body temperature is high, what happens? They are not able to replicate faster. So assertion, the true statement. This is a true statement. Now what about the reason? Is it the correct reason? Fever reduces iron available to pathogen. True, this is also true. As I've told you, they reduces the iron availability to the pathogen, and high temperature the body doesn't absorb much iron. As a result, the pathogens are not getting enough iron. Okay, so this is true, but they are not, the reason is not explanation for the assertion. The fever is stopping the growth of the pathogen, that they have to explain, right? Here the iron availability to pathogen is not a correct explanation. It is also true, but it is not a correct explanation. So here both assertion and reason are true, but reason is not a correct explanation of the assertion. This is the answer. Now let us discuss about the questions, homework questions that are present in your worksheet. Okay, the inflammation response is mainly characterized by pain, redness, heat, swelling. Pain, which means dollar, redness, which is redness, rubor, heat, yes, increase in the increase in temperature is seen, that is fever, we call it as heat, or we can also call it as calor, and swelling, edema. Okay, so all these are the signs of inflammation. So very first option is the correct answer. Itching, sneezing, these are not signs of inflammation, they are allergic reaction. Nausea, vomiting, or muscle weakness, these are not the signs of inflammation. Inflammation has 4 signs: pain, redness, heat, and swelling. Now coming to the second question, what is the role of cytokines? So they are just signal between immune cells. Okay, B cells are sending some cytokines to the T cells, so T cells are sending the cytokines to the other cells. So this is a basically signal connection between the cells. Okay, so these cytokines, second, they are like signals between immune cells is the correct answer. Not destroy pathogen, they don't destroy pathogens. Interferons are the one which kill the pathogens, or we can see other substances that can kill the natural killer cells, secrete some substances that can destroy the pathogen. Now store energy, not the function. Transport oxygen, not the function. So signal between immune cells is the correct option. Now going to the third question, the rapid response to previously encountered pathogen is called. So previously encountered, which means the first response of the immunity is there. Now it is a second response. Okay, the rapid response. So the second response will be always rapid. So this is the second response they are talking about, and previous encounter of previous response is the first one, previous encounter of pathogen. It is adaptive immunity, innate immunity, inflammatory immunity, or immune tolerance. It is called as adaptive immunity. Innate immunity is barriers or any cells that are present from by birth is called as innate immunity. So here we are talking about adaptive immunity. Now going to the fourth question, which cell bridge innate and adaptive immunity? The innate immunity consists of dendritic. The skin basically consists of dendritic cells. These cells have branches. Okay, so they send signals to the adaptive immunity cells like T lymphocytes and B lymphocytes, so that they can multiply faster and they can kill the bacteria, that is, or viral or pathogen that is entering the body. So the answer over here is dendritic cells. Dendritic cells, these are the cells that are present between the multiple layers of connective tissue or epithelial tissue, and they connect to the adaptive immunity, which means B cells and T cells. Now going to the fifth question. This is the assertion reason question. So let us see which is true or which is false. Assertion: inflammation is a part of innate immunity. True. Inflammation is a part of innate immunity. Okay. So this is a true statement, as we have discussed already. The signs of the inflammation that is seen are part of innate immunity. Inflammation helps isolate, eliminate harmful pathogens. Yes, both assertion and reason are true, and it is the correct explanation. So we will go with the first option. Now going to the next question. Question number 6. Skin acts as a physical barrier since secretes lysosome to kill bacteria. So here if you see, this is a true statement. Skin acts as a physical barrier. True. It's the innate immunity. Since secretes lysosome to kill bacteria, this is a chemical response. It is acting. This is chemical barrier. Yes, skin secretes sweat. Sweat consists of lysosomes which kill the bacteria, which comes under physiological or chemical barrier, not in the physical barrier. Okay, the lysosomes comes under chemical barrier or physiological barrier. So this statement is also true. So both assertion and reasons are true, but it is not a correct explanation. So answer is going to be both assertion and reason. So here you have to remember, skin acts as a physical barrier is a true statement. Skin secretes lysosome, then it is a chemical barrier. Then they are talking about lysosomes, they are not talking about the skin, they are talking about the lysosomes. Chemicals are present, then there is a chemical barrier or physiological barrier. So assertion and reason are true, but not a correct explanation. Now coming to the next question, question number 7. Consider the following. Assertion: innate immunity has a memory function. That is totally false. Innate immunity doesn't have memory. It is present from birth, okay, and it prevents the entry of the pathogen to the body. So this is what innate immunity is. So this statement is a false statement. So I have marked it as a false. Now coming to the reason. Adaptive immunity is faster than innate immunity. Again wrong, because innate immunity is present by birth. Okay, and adaptive immunity takes time. Why? Because if the pathogen is entering, it has to recognize the pathogen, it has to form the T lymphocytes, types of T lymphocytes, it has to form B lymphocytes. Now these B lymphocytes will form antibodies towards the bacteria or pathogen. So both statements are false. So here both assertion and reason are false. So the answer is fourth one. Very easy, right? Now going to the next question, question number 8. Identify making A, B, C and given below structures and select the. So this is the structure of antibody that we are going to discuss in our next worksheet. Okay, not here. Now coming to the next. Okay, so we are done with the worksheet questions and homework questions of the 7. Tomorrow we are going to discuss about structure of antibody, and we are going to discuss worksheet 8 and 9. Okay, so we have completed concept based immunity concept, and tomorrow we are going to learn about structure of antibody and types of antibodies, and we are going to discuss worksheet questions. That's it for today. Thank you so much.